Hello, and thank you for watching this presentation by the American Iris Society. Please support the organization by becoming a member. Go to irises.org and click on Join. Thank you.
Tonight, our webinar is by Chuck Chapman, and it's the life cycle of bearded iris. And I won't go through the detailed description of that. Hopefully you've read that, and Chuck will be talking about it in just a minute. Chuck Chapman lived in Ospringe, just north of Guelph, Ontario, Canada, in an agricultural zone 4. He's the owner of ChapmanIris.com and has been hybridizing and selling iris for over 30 years. Some of his iris introductions, such as Forever Blue and Blueberry Tart, are sold around the world. Chuck has had an interest in plant breeding and genetics from an early age. His first hybrid was an apple tree planted in 1949, currently still has a clone of it. Chuck has been studying plant breeding genetics since 1960 and conducting experiments and studying iris biology since then. After a degree in experimental psychology, Chuck worked as a research assistant, then in clinical psychology, working with children and families. For the past 10 years, Chuck has been taking a part-time degree in plant science at the University of Guelph, specializing in molecular genetics and plant biotechnology. So, Chuck, welcome.
Thank you. And this is basically a summary of what we're going to be talking about, going through the different parts of the cycle. The flowers to start with, and then the pollination, seed,
about irises?
seed pods, getting the seeds, removing the germination inhibition, stratification, germination, growing out, planting out, the maturity of the seedlings, summer dormancy, bud set, growth if it increases, winter dormancy, vernalization, spring bloom, and recycling. This is basically a simplified picture of the genetic controls of flowering, and controlling exactly when the plant blooms is very, very important for the life cycle of plants. And basically, all of these genes play a role because the whole thing is a matter of coordinating all these different factors. The only thing I want you to pay attention to is this big pink one in the center, FLC, which is called flowering locus C. And that's a very, very important gene. And the others do all sorts of important things too, but we're not going to look at those or be concerned about it. This is basically breaking the whole thing, that whole picture down into a very simplified form. Basically, we have maturity, bud set, fertilization, and then flowering. And that's basically what we go through with the plants. Now, I'm living in Ospringe, and that we're actually just south of Salem. So I used to claim I was the most northern iris breeder, but I can't claim that. So I can just still probably claim that I'm the coldest area. Basically, we're for zone 4 when, and it might even be, just trying to change something here because I got something sticking in the way of what I'm having to look at. So basically, but Salem gets their peak bloom on May 19th, and our peak bloom here is usually about June 15th, except I got my greenhouse that I grow in. Just something's going wrong here. There we go. I had my greenhouse. I started that about over easily 20 years ago, and this is basically a plastic greenhouse sort of hung inside this big greenhouse, which I'll show you parts of in a minute. And it's gradually evolved over time. So I've been doing this for about 20 years. Here you can see that we're getting a little bit of age on the roof, and you can see it starting to sag down there, and eventually collapsed in a windstorm. And I have replaced it with wood and plexiglass. And now I've expanded even further, and you see the part at the front here is the expansion, and there's the plastic greenhouse at the back. And what I've done is taken this old industrial greenhouse, which came with the property, and this is basically the shape it is when we arrived here. You can see the walls falling down the side. And I'm restoring the first 80 feet of it to be used. And each of those panes of glass has to be taken out of there. The wood has to be repaired and sometimes replaced, and then the glass cleaned and all put back in. So it's quite a job. And this is a picture from a year and a half ago. And this section here is all of the first section that's going to be heated. And right now I'm down to about here with the roof and up to here with the walls. And I find this is very important for a number of reasons. This is the back side of the greenhouse. And the heated part there, notice my nice little chimney there for the wood stove. That's part of my heating process. But the north wall and the north roof, they're not actually north, but northwest, provide no sun during the winter, but they provide area for heat loss. So that's why there's no glass in those sections, just to keep them from doing that. Now, for me, the advantages of greenhouse are quite a few advantages because I'm breeding for cold-hardy irises. But the problem is that a lot of the irises I bring in from the United States, even from Oregon, only about 25% of them actually do well here, and 25 just disappear very quickly. Another 25% sort of, or 50% sort of hang on. About 25% actually thrive. So to breed, I actually have to be able to keep them to bloom. The only way I can get them to bloom and to grow properly is to grow them in a greenhouse. So that's part of why we use a greenhouse. I'm actually increased at about 4 agricultural zones by using cold greenhouse. Gives me a head start on breeding. So when the rest of my iris out in the field are in bloom, I still do some breeding up there, but I have more times to deal with the weeds during that time. A real benefit is I can breed in the bad weather. It doesn't matter if it's cold and wet and rainy outside. I can go inside the greenhouse and I can breed it. So I'm never going to have my bloom season completely washed out by rain or anything else. The SDBs are outside in the field and they are blooming at the same time as the TB inside the greenhouse. So I can make all sorts of nice crosses there to get your IBs. And I also have a lot of my breeding cultivars in a very small area, so I can have them concentrate. I don't have to go running all over the place, because what I do is I select the ones that I want for my stock, plus the ones I bring in that I'm using for breeding, and get them all in that one area. Now I'm just going to give you some examples here of some of the irises that I brought in. I've got more new ones this year that haven't bloomed yet, but these are some that I've had blooming last year. And getting some of the newest cultivars from a whole bunch of different areas so I can use those in the breeding. Mountain Halo over here is one that I'm really interested in and have managed to get a few crosses with that and seeds from it. I haven't had any seeds bloom yet. But Mountain Halo is one put up by Richard Ernst. And I think it's probably one of his nicest irises, and not many people have it because it's not a very strong grower. But the colors on there are very unique, and I haven't seen anything with that much of a contrast in terms of the reverse color. So I'm using that one extensively and hoping to get those genes sort of tied down and make use of them. Just going to quickly go through some other ones that we're working with. The one here, Royaz, that's an SDB. So there's a few SDBs in there in the greenhouse, but it's mostly tall bearded irises. And now I'm going to show you some of my own selects, ones that I've been take and put in the greenhouse to use for breeding, so we can have some pairing up. And I'm not planning crosses for particular individual irises across individual irises, but I planned it in sort of in grouping. So I have a group of orange, for orange hardy, and I have a group of pink for pink hardy. I got some luminatas for luminata hardy, and various things like that. So it's basically a grouping. It's not planning on. And here's some more of my seedlings that I've been working on. That blue one there is sort of iffy. It's not quite as strong and grow as I'd want, but the form and shape and colors on that, and it's immense big flower and tall stalks. So I'm interested in getting, hardy seedlings out of that one. And a few more of the ones I'm coming. The one on the 14-5-61-1, extremely hardy, very early bloom, and that's one that I will be introducing because it's just vigorous and strong grower. And We don't have very many of those colors that actually survive well in cold climates. Again, working on the orange ones and luminata, fancy luminata one over here. Color, and there's working on my hardy orange variety. This one's not quite as hardy as I'd like, but still hardier than most of the ones up from Oregon. So that's being used extensively in breeding as well. Canadian Rerun, which is a very strong rebloomer and has tremendous genetic potential. So I'm using, and Ragtime Singer over here is actually a color changer. So it changes color as it blooms through the season, depending upon the sun and so on. They have many different shades of presentation. Now, irises actually have 3 sets of genes. Now, when you think of genetics, people think of the nuclear genes, which are, you get half of them from each parent, and that's, what you're doing when you're breeding. But you have your mitochondria genes as well, and the mitochondria, showing you up here, sort of the red part of the cell, has its own set of genes, and the plastids, and it's basically, people know them as the chloroplasts, is the green here. But I mean, they're also the chromoplasts, which are involved in the color and starches, starch storage and other things like that. So, but the mitochondria and the plastids only come from the pod parent. Now, I'm using my own seedlings primarily as a pod parent, and the other people's introduction primarily as a pollen parent until I get enough information to know that the one that I brought in can be cold hardy, and therefore I can trust using it. Now, factors that determine cold hardiness are located basically in the mitochondria, and those only come from the pod parent. And the direction of crosses can make a difference. I keep getting this argument. People keep saying, well, I make crosses both ways and it doesn't make any difference. Well, if both parents are similar in traits, then it won't make a difference, but it does make a difference. Now, one of the courses I took at the University of Guelph a few years ago was a 4th-year breeding course, advanced hybridizing and breeding. And in the university is doing a lot of agriculture breeding. So, asparagus, wheat, rye, corn, a whole bunch of different things. So we got a chance to meet with and talk with all sorts of different breeders in the university. And I told them what I was doing with my irises. And basically it's also basically on the literature, because the literature has shown this very clearly that, your cold hardiness comes from the mitochondria part of this. And they all do the same things. All these hybridizers universities, they're doing all these advanced breeding programs. When they bring in seed or a plant from a warmer climate, they breed in a greenhouse, like I do, and they always use the ones from the south as the pollen parent and their own stock as the pod parent, basically because of that same thing. Cold hardiness has to come from the maternal parent. So that's a very, very important part of the breeding program. Now, one of the first things you got to do if you're going to do breeding is to actually, make some crosses. That's a picture of Sylvia there, and this is quite some time ago, and she's, I think, about 5 years old right there. And she saw me doing it in the garden, and she had to try it for herself. So she's actually made some introductions too. And I'm right there. It's late in the season, and had trouble with weather. So what I did is I collected the flowers and kept them inside overnight, put them in a cardboard box there, and took them out in the morning so I could use them. Now, basically, you need to know what the parts of the irises are if you're going to do some breeding. Now, the style arm part in here, and then you've got your stigmatic lip and your crest, and your anthers down in the center here. But basically, here's a close-up of your anther, showing it there. Here's your stigmatic lip with it sort of pulled back so you can see that lip, and that lip is actually sticky. And what you got to do is get the pollen onto the stigmatic lip, and that's what you have to do in order to make a cross. After you make your crosses, then you end up having your seed pods, and that's a collection of seed pods in the greenhouse, because I do a lot of my breeding. Like I think about 75%, 80% of my crosses are actually made within the greenhouse. And so everything is loaded down with seed pods inside there. I'm showing you a couple of interesting pictures here. Now, the one here on your left, you have 4 pods in the terminal. So I had 4 flowers on that terminal of that plant. And that's one of the reasons I enjoyed that plant. And every single one of those terminal flowers formed a pod. So here you got 4 pods in a terminal flower setting. And over here I have a set of 7 pods on 1 flower stalk. So the greenhouse gets used really heavily. Now over here we have what a seed pod looks like. On your left-hand side, you have what a seed pod looks like when it's ready to harvest. Actually, I was a little past ready to harvest, but I took that picture because we wanted to show you what happens. The seed pods, as they get mature, turn brown and they start to crack open. And generally, I'm picking them when they're brown and just starting to crack open. And I'm showing you some variations in sizes here because you have seed pods come in all sorts of different sizes and shapes. Those number tags there are what I use for my for identifying the pods. And I put those tags on there, write the numbers down in my stud book, and then enter them in the computer with the, so I have each one of those numbers. I can refer to what the pod and the pollen parent are and record that information. Occasionally you can get bee pods, and on your right you have what a bee pod looks outside. On the left is what it looks like when you cut it to the center. And we have lots of bees. That center part there, we have 28 bee pods, beehives on the property, and those guys are busy and they're around all over the place. And those are irises that you just see in front of them there. But you do get these bee pods, and they don't, and the air pods, and they're just basically empty. So be on the watch out for that. So sometimes people get excited about getting a pod just set it by itself in the garden, but unfortunately, a lot of times they're not really, useful. And when you have to move irises around, you don't want to be planting seed pods with them. So you cut the seed pods off and stick them, in this case, in a bucket of water. Various people do different things like sticking them in sand, etc. But a lot of people do the same sort of thing. And then you have them all in the same place. When they start to turn brown, you can harvest them. On the right is what it looks like when you open a seed pod and you see all the seeds inside it. Seeds come in various colors, and I'm showing you a selection here from sort of like an ivory brown right straight through to a dark chocolate brown. And again, on the left, some variations on the different sizes of seed pod. That one seed pod there that I'm holding in my hand is just absolutely huge. The seed pod's probably about 4.5 inches long. And actually, the size of the seed pod doesn't guarantee that you'll have a lot of seeds, because sometimes you open a big pod like that and you only have maybe 4 or 5 seeds. But usually you can get up to about 100 seeds in a pod. And when you harvest the seeds, they have to dry before you do anything with them. So here it is out in the Quonset hut, and I got them all out there. And that's about a third of them, because I usually have about 3 tables set up, and that's just 1/3 of them. Now, before you can actually get the seeds to germinate, you have to remove what's called a germination inhibitor, which is involved in the seeds. And usually to do that, you soak for a week and you rinse on a daily basis until you get them removed. Now, what I did, I took 50 seed pods and I soaked them for a full week just without rinsing, and then took 10 milliliters of that and used it on mustard seed. And on the right-hand side there, you see the ones where after 2 days, you have 1 of 25 seeds germinating, whereas on the other side, you have all the seeds germinating. So that inhibition chemical is there, and it's actually abscisic acid. And one of the things that it's known for is it's part of what causes, involved in the leaves falling in the fall. But it's also responsible for inhibiting seed germination, and it's one of its main products of doing that. So we have to get that out of the seeds before we can actually do anything with them. And if you don't normally if you plant them in pots or put them outside, the fall rains will generally remove that from them, particularly if you have a lot of fall rains. And I have seen a number of hybridizers who will actually put a water sprinkler on their seed pods in order to help remove that inhibition from them. And there's a few people who will put them on, tie them up in pantyhose and put them in the back of their toilets. And that's fine if you have 1 or 2 pods, but when you have about 400 pods, 500 pods like I do, you don't have a toilet tank that's big enough for them. And what I do with the seeds, I tie them up into pantyhose and, you know, tie them off there. And then I put them into a shoebox with damp peat moss and put them in the fridge for the stratification period. Now, stratification is an interesting process. It occurs best at 4 degrees, and it has to be wet or moist, and it has to have moisture in air and cold in order for the stratification to occur. Now, I've got a chart there that shows you what I call chilling hours. And as you can see, the time, you know, between 37 and 48 degrees Fahrenheit, or you know, around 4 degrees Celsius, you have your maximum chilling hours. And it's not the colder the better, because when you're just at freezing or below freezing, you do not get any chilling hours. When you go above a certain time, you actually are removing chilling hours. Seeds need to be in that cold period for about 8 to 12 weeks in order to germinate. And when they've done that, they come out and what I call my happy seed pictures. And you see that they're all nice sprouted there. And I think that's when seed pod had about 100% germination. So that's what you should be getting after that time. At this point, I plant them up in little pots, and I'm showing you sort of the stages of growth, with the left-hand side being just newly germinated, and then you've got, you know, 2 different time periods there when the seeds are sort of maturing in these pots. And they're growing on inside the greenhouse. You see them on the left-hand side being inside the greenhouse. So that's in the heated part of the greenhouse, or the insulated part. And then then before you put them outside, you have to acclimatize them. So you have to be outside and get used to the weather and direct sun and so on. So that's showing you on the right-hand side, getting acclimatized. And then you go to plant them out. And that's basically what a seedling looks like at the time you're actually putting them out. And when I get them the head start inside, I get them outside on the June 12th. I actually probably could start planting a little bit sooner, but that's the field on June 12th. And there's, you know, like 2 months later and 4 months later, you're seeing the growth that we're getting on those irises. So what I want to point out here is that when you see irises sitting in dormancy, people kind of assume it's a heat dormancy, and it's not. And I'll talk with that in a second. Because these seedlings are actively busy growing when you have other plants that are sitting in dormancy. Now I'm showing you here some of these. These are some of the seedlings out in the field. And you can see they're already having increases, so they're growing well. And when they have increases, I know they're going to bloom next year, and I'll talk about that in a few minutes. Excuse me. The one on the right-hand side is a standard dwarf seedling. And this is rather exceptional because we have about 8 increases there that are looking like they're at maturity at the same time. And the only other time I've had a seedling that do that, that was Forever Blue, and it's Maiden Blue, and it had 8 blooms on its first thing because it had such strong increases. So I'm watching this SDB seedling really interesting, because on that cross I have a numerous number of SDB seedlings that are actually showing very similar increases. So just fantastic increases and lots of potential. So this is basically sort of like your different sizes. You know, as the plants mature, you're looking at the different sizes as they're getting larger. Now, there is a maturity point with irises. Now, all plants have to have maturity before they can actually get to bloom size. And sometimes you don't really notice that, and it's hard for you to tell. Irises, it turns out that it's actually the number of mature leaves, and it's a number of mature leaves counting the ones that were there as well as the ones that are there. And the ones that were there leave scars on the rhizome. And scars that you're looking at are the ones with the little holes there. You see on the right-hand side, I'm showing a close-up, and you see the holes. And that's where the veins were connecting through from the leaves to the rhizome. And that's the part that's the scar. And that's what you count them. And you have to count those up. Here I did a sort of like a review a few years ago. And this looked at the number of mature leaves when a plant is blooming. Now, it varies. And I found it varying from between 9 and 16. Now, this is a little bit deceptive because I counted them when they're actually in bloom, and it turns out there's a couple of leaves that are on the flowering stalk that you are not really part of the maturity. And so basically on some of those, you might want to take off two. But basically, the idea is showing you is that there's a lot of variation from cultivar to cultivar. Now, I'm talking about here apical dominance. This is a very important concept because it involves when you see the increases and, so when the plant gets ready, it increases. Now, basically, an iris rhizome is a modified stem. So when you look at the tree branch on here and you see the stem and the leaves coming off at different nodes, on the rhizome, each of these sort of rings are a leaf, and your nodes are there in between. And if you notice on irises, they're always sort of paired on opposite sides of the main rhizome. You don't get increases starting in the center, you don't get them on the back, they're just always on the side. But when the iris is producing leaves, the apical meristem, growth point at the top, and I'll show you a couple more slides on that in a second, actually sends a chemical, plant hormone, down the stem, which represses and stops the increases of growing, and this is known as an apical dominance. And there's showing you sort of like a slide of basically the apical meristem, which is right at the top of the stem. It produces your leaves when you're in your vegetative state, which is a leaf growing state, and your new leaves growing, and your lateral buds there are growing in the the side branches, and those lateral buds are actually in irises are the increases. So that's where they are on each side of the stem there. To get to the flower development, you have to have that apical meristem go from a vegetative phase to a reproductive stage or the floral stage. And to do that requires a particular signal. And on irises, I've referred to this as the bud set, and I'll show you some of what involved in figuring out what that was. I'm showing you this picture here, and this was a picture done through a microscope by Marion Scholl in 1923 and published in the American Iris Society Bulletin. And he's the first one who actually looked at when, bud set occurs. And he's looking at this in late fall. And again, looking at all of these different genes that are involved in controlling when the plant flowers. It's all important for plants to control when the timing of this flower, because it's very critical for its survival. It has to bloom when the rest of the species are in bloom because it has to have partners. It has to bloom at a time when pollinators are available. And don't forget that many plants are competing for the same pollinators. So you have to be blooming at a time when you can actually attract pollinators. You can't be blooming at a time when there's something very much more attractive in bloom that's pulling the pollinators away from the flowers. So you have to be able to get your pollinators to do it. You need time for the seeds to mature. So that's part of the timing of the flowering. You have to have the energy for the seed maturation. You have to have suitable weather for pollination. If there's extremely rainy weather at that time when you're flowering, then you're not going to get any pollination. So that's part of what has to be sort of gathered at the same time. If a plant blooms at the wrong time, they're spending plant energy, and plants out in the wild actually have to compete very strongly with members of their own species and members of other species, and it has to be competitive. And if it's when we get an iris blooming at the wrong time, for instance, rebloom, it's basically the failure of one or several of these genes in order to control the time when it's blooming. Again, we're looking at maturity, bud set, fertilization, and flowering. So now we're looking at the bud set. I'm going to talk a little bit about dormancy here. Now, people think of dormancy as being a plant sort of goes to sleep and it doesn't wake up until spring. And that's called endodormancy. And I'll sort of refer to that in a few minutes. But basically, we don't really get much of that in irises at all, especially in the modern hybrids. Ectodormancy is basically the plant doesn't grow when the growing conditions are not suitable. It's too hot, it's too dry, it's too cold. The conditions are just too dark. It just doesn't have the conditions there, which it needs for the growing. So basically, on a cold day, it doesn't grow. On a warm day, it will, because it's not in deep dormancy or endodormancy. And paradormancy is the one that I'm sort of going to be talking about next. And basically, for an iris, we have paradormancy, which is basically the time between it reaching maturity and the time we get bud set. Now, when I first started looking at that, I was looking at these pictures that we see in all introductory biological texts, and they all show irises as being a long day plant triggered by days above 12 hours of daylight. And I look at this and it didn't make any sense to me. And one of the nice things about biology texts is that they usually have a reference, this information was gotten from such and such a point. But when you look in the modern biological text, you see this picture, you see this story about irises, and there's no references. So I figured, I want to find out the reference list. I want to read the original study. I always like looking at original study because I'm looking at the methodology and how they do the study to find out if they have actually control for all the factors that they need to control of and actually have done the experiment properly, and the experiment actually says what it's, the conclusion that they reach is justified by what they have done. So I went to the University of Guelph library and pulled out some textbooks and, gained no reference. I went another couple of decades back and pulled another biological textbook, same sort of picture. I gained no reference, and I finally ran out of textbooks in the University of Guelph library because the old ones are actually in storage. So I had them pull out the old ones in storage, and they kindly did that. They bring it in to me, and I read it, and I gained no reference. I finally went back to about 1920, 1930, something like that, and found an original reference. And okay, of course, I didn't have the study there, so I gained another, put it in for a request for it. I have to, find some place that has this article and get a copy and read it. And it turned out to be done by Dutch irises, and they did not control properly for lighting and temperature and any other thing. So actually, the only thing to look at was light, and so you really couldn't trust that study. When you look at when irises are peak blooming, we find the range all over the place. In Los Angeles, California, peak bloom is about the end of March. Modesto, middle of California, usually about the middle of April. Salem, California, May 19th. For me, for Hamilton, which is just like half hour south of me, peak bloom is June 3rd. My peak bloom is June 14th. And Thunder Bay, Ontario, June 21st. But we get the opposite when it comes down to rebloom. Rebloom for me, for the tall beard iris, it will start in late July, whereas Modesto, California, for instance, starts mid-October. So basically, it's the reverse. So something's going on here, but it's not daylight that's controlling this. There's some research that was done on bud set, but there's not a lot of them. There's Marion Scholl, which I talked about, did his stuff in 1923. The fellow in Germany did a study in 1932, and he found that bud set was occurring in June. And they did this sort of destructive by, taking it and dissecting it and looking inside until they could actually, see what was going on. Another article by Megan, I don't have the location for that. They found the bud set in September. And then a group of people in 1967 did a study, and they found it in August. So we're getting these bud sets all over the place, and it's obviously not photoperiod, which is triggering this. So photoperiod is not doing the same thing at all. Those people who want to know what their weather history and their history, and this is important for for looking at for bud set temperatures and all. I sort of probably should have put this slide a couple later, but I use Weather Underground, and if you notice, it has it says history. And when you're clicking on history, you can see the history by month and for a year and look at your weather data to find out what you had. Now, one of the nice things about internet is that you can coordinate with people. So in a number of groups, this was even before we had Facebook, Facebook as a group process, and this was using like Google or some other sort of group. And so I picked immortality as it's a rebloomer. It reblooms early. And we're trying to find out when it reblooms and what is the trigger for reblooming. So I had people all the way across the United States tell me when your immortality is blooming. And I had a sneaking suspicion it was weather. So I go to Weather Underground, look up their data and look through the information that they had and find out. I looked at my own weather charts because I keep my own weather charts, you know, on every day I record my own weather here. And here's a picture of immortality, July the 4th, July 22nd, and July 26th, basically in paradormancy. It's sitting there, it's a maturity, the leaves are not growing, nothing's happening. Then we have this weather. And, you know, here between July 16th and July 23rd, we had 6 days of a temperature row between 15 and 20 degrees C. Now, I had a sneaking suspicion that these are temperatures from all the information I've received from various people and looking at the data and sort of coordinating and looking at it and, you know, examining it closely. I had a pretty good idea that that's what we're looking at. So I had those 6 days, you know, and then on August 1st, you see this over there on your far left-hand side. That's what's called a pregnancy hook. And that's the first sign of a new leaf growing and a little bit of waviness on it there, showing you, and that's showing you the bud set's coming. There we have August the 3rd, 2 days later, and you can see the bud starting. And August the 8th, you can see that actually the flower stalk is coming. So we had the bud set, and off we go. Now, a little bit of a caveat here, that all of these studies, mine and all the other studies, they were field studies. So we don't have any control of the weather. We don't have any control of the temperature. We don't have any control of the you know, the nights of rain, of number of hours and so on. So basically, all of that information has to be confirmed by a study in a growth chamber where you can control exactly the temperature and exactly the lighting that it receives, number of hours, the intensity, and so on. So you can control all those factors and actually evaluate it. Immortality and Canadian rerun are both parts of these rebloomers that'll rebloom as soon as they have bud set. Another indication of bud set is we have removed the apical dominance. And when you do that, increases start to grow. So I have two pictures here of increases starting, one where it's just starting, one where it's a little more advanced. And actually in my own hybridizing program, what I like to see is an early start of increases, which meaning that those plants probably have a colder bud set temperature, because the bud set temperature for immortality is just immortality. And of course, there's variations from that. And that's basically what I've used because that's what I determined with, because it was obvious. So basically you get the increases going from there. Now, another factor that's involved is dormancy. Now I'm talking about endodormancy. And over on the right-hand side there, you see a picture of Iris aphyla. This is actually a picture in spring, but it shows you, what I'm talking about. Iris aphyla is a true endodormant plant. It goes dormant in winter and stays dormant all winter, regardless of the weather, and doesn't grow again. I took a piece of Iris aphyla in the fall after it had been triggered into dormancy, brought it inside, put it in a growth table, and watered it and gave it light and watched it all winter long, and it did absolutely nothing. Nothing, all winter long. Come spring and summer, I figured, okay, I'm gonna put this out in the greenhouse. I took it and planted it in the greenhouse. Again, nothing, because it did not have the vernalization needed to bring it out of its hybridization, out of its endodormancy. Now, Iris aphyla has been used in quite a bit in iris breeding, and there's quite a few of the rebloomers that actually trace this back to Iris aphyla. Now, I was talking about vernalization and in terms of flowering. Well, the vernalization gene is also used in winter dormancy, and there's a lot of the same genes that are involved in flowering are actually involved in your endodormancy. But what's happening with Iris aphyla? The vernalization gene is tied to the dormancy of the plant, and does not have any effect on flowering. So basically, in Iris aphyla, it's a recessive gene. As soon as you cross Iris aphyla to another plant, you now have those genes, you have removed the vernalization genes needed for flowering, and you get sort of like a mixing of genes. Now, on the bottom there, I have a cross of Iris aphyla with Forever Blue, and this is a very, very strong rebloom. And that picture is actually in late August. As you can see, that row of plants, and just about everything is reblooming because we have those triggers that are mixed up there and has provided that fall bloom. Fertilization is needed for flowering, and you have to watch that very carefully because vernalization is a little bit tricky. People think that vernalization is when you have the actual cold period, but it's actually not. Vernalization, again, that shows chill hours, maximum at 4 degrees C or about 40 degrees Fahrenheit is when you get your maximum. Below that, you have less chilling hours. Below freezing, you have no chilling hours at all. So your plants can be sitting out there, you know, under snow and at 0 degrees, and they're not getting any chilling at that time. Now, you have to have, and apparently with irises, you seem to have somewhere between 450 and 600 chilling hours for it to bloom to get proper fertilizations. And it's rather deceptive. Now, this is a chill map of the United States, and it shows you sort of like the different chilling hours. And this is why you don't get bearded irises blooming in Florida, because you just don't get the chilling hours. There's actually one exception to that, which I found someone who lives basically on an island just off the west coast of Florida down here on a tiny island. And he's far enough away from the mainland that he actually gets enough chilling hours so he can grow bearded irises there. And he's the only person I've found in southern Florida who can actually grow bearded irises. Now, what happens with vernalization? Now, I talked to you before about the flowering locust seed. Remember that nice little pink gene in the center of your flowering thing? Well, vernalization actually does is it removes that gene and it activates it. Now, for any gene to be active, it has to be available to be read by the cell mechanism. In order to be read by the cell mechanism, it has to be unpacked. So you're seeing on there where it says FLC active on your left-hand side, and you see it shows it being unpacked here, there, and available for being read. What vernalization does is it packs those genes. So FLC is now packed, it can't be read, and it can't be used. And what things that the flowering locust seed does, it actually prevents the plant from flowering. So when it's packed like that, it is removed from flowering. Then it can't prevent the flowering. So when you have your vernalization, when you get flowering, and this is a seed bed from a number of years ago, that's showing nice spring bloom. But one of the things that happens with that is that it's an epigenetic gene. That's an epigenetic change. And the thing with an epigenetic change is that it's passed on from parent to child. So basically, once the vernalization is being, it should pass on to all the children, but it doesn't. There's a mechanism there for reset. I'm not sure what that mechanism is for reset, but it's there, and it goes basically from the mother plant to the daughter plant. But what is happening with most of those rebloomers and rebloomers of all types seem to have that vernalization is not reset. So it contains there, and when you get to the bud set, you get the bloom. But I have my whenever rebloomers like Forever Blue, Blueberry Tart, and those in that grouping, what happens is that they bloom as soon as they reach maturity. So they seem to have both, have not reset the vernalization and also have not reset the bud control. So they bloom whenever they reach maturity. They don't seem to need any vernalization and so on. So that those reset is important, and that's an important part of resetting the plant. And for that, there's Forever Blue and Blueberry Tart. And there's basically a summary of what we've gone through. So I've just sort of covered all of that. And I'm just going to put that slide back up there. And I didn't hear any questions during the thing, but I'm hoping there's some questions now, and I hope I didn't make it too confusing.
Yeah, Chuck, I have one question from Anna, a couple questions actually. One is, why does the iris produce empty seed pods, and what triggers it?
I really don't know that one, and a lot of people sort of wonder about what's going on in there. But the formation of seed pod is actually under a separate genetic control from the seeds itself. So it gets triggered by some sort of mechanism. It's part of the, the mechanism. And you think they're tightly connected, but they're not actually as tightly connected as they should be. And one of the things I found, though, is that I often get those air pods on flowers on plants where I've actually used them in hybridizing. So the plant's been shaken, or it's been moved, or I've sort of brushed against it, and for some reason there's more air pods set on that. So the more you actually got physical touch, or it's being moved, by whatever, it sets seed pods. So it has something to do. That has something to do with it, but the actual biological mechanisms, I really don't know.
Okay. And then the second question from Anna is, do cold hardy irises perform better in warmer climates? And I'm not sure, are mitochondria produce more energy in warm climates for better performance?
Well, the mitochondria are basically your energy parts. Now the plastids are forming are making the sugars and so on, because they're produce the sugars. But the mitochondria are the ones that sort of are the warehouse of handling the energy, deciding where it's going to storage and how it's sort of used and the whole thing. And they have a number of genes that are involved in that. And they basically seem to operate wherever they're, it doesn't matter what the climate are, they're going to produce, if they produce more energy, they'll produce more energy in any climate. Now, one thing I have found that's very interesting is that my cold hardy irises that I produce, when they are grown in warm climates, seem to also do very, very well. And so it seems to be just a general hardiness, that's involved in that. Now, I haven't seen any yet so far, I've not seen any of my hardy talbot irises that don't perform well in the southern United States. But part of the problem is that a lot of people in southern states are not buying from me, because a lot of people are intimidated by buying from Canada. But I have a process set up which makes it super, super easy, because I just take my orders and I have one shipping date. And so I bring them all, I get all the irises ordered, I get the agriculture inspector to come in. So it doesn't matter whether I got one order or if I got 40 orders, it's the same price to me, same cost to me to have the inspector come in there and inspect all 40 plants. So the phytosanitary cost of inspection gets spread across all the orders. I pack them all up and I take them across the border to Lewiston, New York, where I use the United States Postal Service and mail them all out. And you get those suckers within a day by mailing them. And I made it super, super easy. So there's no excuse. But part of the problem is that my cold hardy irises don't have quite the same form and fanciness of some of the southern ones, which is why I bring the southern irises up into my greenhouse in order to breed, in order to get that. And I'm now getting more cold hardy irises, better form and better shape. And if you tried these guys in southern, the more southern climates, I'm absolutely sure that they're actually going to do well and be just as hardy in the southern climate as they are in the northern climates. It seems to be a general hardiness. It doesn't seem to be specific